and this is rough endoplasmic reticulum. We can tell it's rough endoplasmic reticulum because it has ribosomes clinging to the surface, unlike smooth endoplasmic reticulum on the other side of the nucleus, which doesn't exhibit this feature. So what does a cell look like when it's not appearing in an idealized cartoon drawing? In order to understand that a little bit better, we'll need to know a bit more about the light microscope and how tissues are prepared for visualizing under the light microscope. So the light microscope consists of a frame which holds everything together. This is the stage, and it's on the stage that the microscope slide will rest. There will be fine and coarse focus knobs that will enable you to move the stage up and down and bring the lens closer or farther away from the objective. And there will also be small control knobs here that will enable you to move the stage back and forth under the objective so you can see different parts of the tissue. How the image is formed is a light source, comes from the lamp, goes first through an iris diaphragm, as you would find in a camera, and then reaches a condenser which focuses the light on the plane of the slide. After it goes through the plane of the slide, it reaches the objective lens, and light from the sample will produce a magnified image within the microscope. After the image is generated within the microscope, it passes through a second lens, which is the ocular lens, which is just down from the ends of the eyepieces that you will be looking through. So lamp through the iris diaphragm, condensed, the light beams are condensed onto the surface of the slide, passes from the slide through the objective lens into the microscope, then through the ocular lenses to form a visual image on your retina. Depending on the objective that you select, the image will be magnified 10 times by the objective lens, or 40 or 100 times, and then 10 times again by the ocular lens in the eyepiece. So if you're using the 10x, the objective that magnifies the image 10 times, the image will be 100 times larger than you see on the microscope slide when you're looking at it through the ocular. So it's magnified 10 times by the low-power dry objective, the first lens, and 10 times again by the ocular lens that you're looking through right at the eyepiece. Another example, if you're looking at a slide with a 40x objective, the image will be 400 times larger than it appears on the slide, 40 from the field objective and 10 from the ocular objective. So those are the mechanics of the light microscope. In order to be able to see something in the system with light passing from the lamp through the slide and two sets of lenses, we have to prepare tissue sections that are so thin that the light can actually pass through them. The tissue sections for light microscopy slides are somewhere in the range of 5 to 8 micrometers thick. And a micrometer is a millionth of a meter, which is a thousandth of a millimeter. To give you some idea of how thin that is, one red blood cell is 7 micrometers thick. So a very thin slice. So the first stage in preparing tissue is to fix it. And so a chemical fixative is added that kills the cells and stops the chemical reactions. The most important chemical reactions would digest the cell itself, a process called autolysis. Then after it's fixed, it's embedded in something that will make it rigid enough to be cut by a microtome. So that's why you put the steak in the fridge before you slice it into fine slices in a recipe. How things are sectioned has a profound effect on how they actually appear on the slide. So let's take a simple object. This is meant to be an egg. So this is an egg, and this is a transparent egg. So let's imagine for a minute that the white is the cell cytoplasm and the yolk is the nucleus. So how we see the egg on a slide would depend on the angle in which the section was cut. So first, let's imagine that there were two cuts, the two green lines on the left, sagittal sections through the egg, and they're right in the center of the egg. So on the regular egg, on the transparent egg, and then if we take that section, turn it 90 degrees, then the green section on the right is how the egg would appear in the slide. Similarly, if instead of doing what would be similar to a sagittal section of an egg, we would do a coronal section of an egg, we would get exactly the same thing. So slicing the egg, 2 cuts, turning the egg 90 degrees onto the slide, and again we see cytoplasm and yolk of the egg. If, on the other hand, we're not right in the center, we're slightly off the center, actually this is about 1/3 of the way off the center, and we look at the transparent egg, we'll be cutting a section through the white and through the yolk, but not through the center of the yolk. So we might miss, for example, a nucleolus if that were in the center of the nucleus, and we would have something that doesn't look exactly like the idealized version of the cell. If we move even farther, we'll miss the nucleus altogether, and so in this case, the egg, representing the cell, will just look like a chunk of cytoplasm. So not every cell will look entirely like the idealized version, and on a particular tissue slide, some will look like the cells you expect to see, and others will look like something else again. And that again is how we train our eyes to see, and how we train them to see is because we're thinking about what we're actually seeing. This time, instead of doing a sagittal or coronal section in the long axis of the egg, we're going to do an axial or transverse section. And again, this is something that could happen to a range of cells within a tissue. So this again is centered nicely, so we'll get egg white, egg yolk, which would be cytoplasm and nucleus, as it would be right in the middle of the cell. If we're slightly off the center, and we do what's called in histology a grazing section, then we'll get less of the egg yolk, more of the egg white, a different, smaller part of the cell, and again it won't look like the idealized version. And finally, if we just do an axial section of the tip of the cell, that is all we'll see on the slide. And that's one reason why every cell on a slide, a tissue slide, will not be identifiable as something that you would recognize, and why all the features that you expect to find in a cell may have been left out in another section. We've looked at the basics of light microscopy. We've sectioned the tissue, and now we have to stain it. So after sectioning, we'll have a very thin slice of tissue, but it will be colorless. So how do we bring it into some form where we can actually see the tissues? We'll stain with 2 stains, a bright stain and a counter stain, and because of their chemical properties, the stains will pick up different features of the cell. Hematoxylin is a basic dye, and as a basic dye, it will bind to acidic cell components and stain them purple or dark blue. Examples of things that stain purple or blue using hematoxylin would be DNA and RNA in the nucleus, other carbohydrates, and certain features of the extracellular matrix outside the cell, and we'll see that with cartilage. Eosin is an acidic stain, and so that's going to bind to basic cell components. So cytoplasm, filaments, the plasma membrane, collagen fibers outside the cell, and so that will stain things orangey pink. So if we apply hematoxylin to our cartoon cell, we'll have a cell that looks like this. So the nucleus will be purple, lots of DNA. The nucleolus will be dark purple, lots of RNA synthesis going on. Then the basic cell components, the dye will bind to them, and so these features of the cell will be pink. And again, that's cytoplasm, also muscle filaments, intracellular membranes, and things outside the cell, such as collagen fibers. If we use both stains, we'll have a cartoon cell stained to look relatively similar to the cells and tissues that you'll be seeing in a moment. So hematoxylin staining acidic components blue, eosin staining almost everything else pink. So for a quiz question, when hematoxylin and eosin stains are used, and these are the stains that you'll be seeing, Which of the following structures would bind hematoxylin?